First of all, thank you for the opportunity and allow us to show in some of the small projects we have currently at the Department of Food Science and Microbiology and Fermentation at KU. So the small story I'm going to talk today is about the relationships between the gut microbiome and lipoprotein distributions and in healthy subjects in the Copenhagen area. So basically, lipoproteins and the cholesterol that they carry, as you may know, they are important precursors of hormones and bile acids important for our body. And alterations in the metabolism of lipids, often called dyslipidemia, may represent risk factors for the development of atherosclerosis and cardiovascular diseases. And in recent years, perhaps in the last 3 to 5 years, there have been some advances in metabolomics that have allowed us to improve the resolution of mapping lipoproteins in different subclasses, because when we're talking about HDL and LDL, it's not only one whole particle that we should look at it, but actually we're looking at different subclasses like the one you can see down in this figure over here. And the reason why we are so interested in the different subclasses is because the different sizes of particles we have, they may tell us some more information about the type of risk that we're having in developing some kind of cardiovascular diseases. So this is the case here of HDL, for example, where the larger particles they are, the more they tell us information about the lowering the risk for development of cardiovascular diseases, while small particles of HDL, like for example, the one you can see here, basically doesn't really tell us anything about the risks. In the other side of the spectrum, you have the gut microbiome as well, that within the last 10 years, it has been linked to disease progression, a lot of metabolic and chronic diseases, and most of the studies have been done. We have extensive characterization in type 1 diabetes and type 2 diabetes. And in a lesser degree, research has been done in relation to CVDs and the distribution of these lipoproteins in the body. So there are a lot of unknowns that still we are trying to research. And some of those ones, at least for the purposes of the project that we're working now, are like what kind of relationships between lipoprotein profiles, distributions, and current recommendations of cholesterol can be established. Also, we have a lot of limitations, limited knowledge in relation to what is going on within the gut microbiome and the lipoprotein distribution axis, and whether there are some gut microbiota factors or important molecules that are produced by the microbiome that can be essential for lipid metabolism. Part of these questions that we're trying to address them in the Counterstrike project, where we have collected samples from healthy individuals in the greater area of Copenhagen, from different ages, ranging from 20 to almost 90 years of age, different BMIs, and from them we have mapped the lipoprotein distributions in the plasma, and sequencing a lot of data from, for example, 16S gene amplicon, from short-chain fatty acids, and shotgun metagenome from a subset of samples. So the first part of the results, it looks like this. It's a very complicated figure, I think, and it contains a lot of information, but this is what represents the lipoprotein distribution of these 260 individuals. If you see the upper part of the figure, this is where it represents the current recommendations about the levels of HDL or LDL or total cholesterol that the individuals should have. So the darker the color basically tell us that they are out of the recommended ranges. If we look to the left side, this is the different lipoprotein subclasses or species that we may have within, for example, HDL or LDL or VLDL and so on. And somewhere here we can have some information about the different phenotypes where the individuals may cluster according to the different profiles that they may have. So If we look at the middle cluster of people somewhere here, which is something called cluster 1B, for example, these are people that in general, they have values of HDL that are within the recommended levels. What we can see here is that the total HDL is basically driven by large particles of HDL and something that we call that 2A and 2B. And this is within the last probably 20 years, there's not too many studies that actually have looked at it, but at least the 2 or 3 that we can see there, they have been associated. The higher the concentration of 2A and 2B we have, the less the risk of developing cardiovascular diseases can be seen, at least. And if we look at the right part of this heat map, then we have individuals that have a high concentration of total cholesterol and LDL in their plasma. And this is also driven by larger populations of subclasses of LDL1 and LDL2, for example. So even though we have subjects that they're apparently healthy in this cohort that we have recruited, you can still see that maybe there are some phenotypes that may not have a very healthy LPDs profiles according to what the literature says. And this is the case of the cluster here, C1A. Interestingly, 25% of the total variance of these LPDs distributions are explained by variables like age, BMI, and sex. And I think it's interesting, BMI here, because it's not only BMI per se, but perhaps this tells us a lot of information about the lifestyle that these people is having. So again, I'm going back to cluster C1A that lies somewhere here. And apart from having a total high cholesterol and high levels of low density lipoproteins, they also have a higher BMI. So there's a lot of interaction there that at least that we are not catching that yet, but we're trying to screen the whole, at least the cohort at the moment. So, but we can go also to the gut microbiome and see what's going on there by sequencing the 16S gene. And what we can see is we can discriminate the different clusters that we can see from the LPD distributions. And what we see is a transition in the abundance of different bacteria phenotypes that are common from the people in Copenhagen. For example, the people that has the more unhealthy profile, for saying that way, which is cluster 1A, they have a higher proportion of Lactobacillus in their gut microbiome, for example, and the more healthy profile you tend to have, whether it's cluster 1B or 2, then you tend to have a high proportions of Ruminococci and Christensenellaceae and Bifidobacteriaceae, which is typically what we know as a very good guys to have in our gut. This is only taxonomy, but we can also analyze that all the genetic information and metabolic potential that the microbiome of these individuals have. And by doing that and sequencing the total genomic DNA of the bacteria, what we observe is a loss, basically. If we look at here the clustering, the higher we have, for example, if we go to cluster 1B, which is a very healthy, relatively healthy profile, then we have a loss of functional genes that are associated with primary and secondary metabolism. And some of those ones were very interesting what we observed, because it included the metabolism of short-chain fatty acids, as well as the biosynthesis of vitamin B. And if you remember now, the cluster 1C1B was those phenotypes with the lowest BMI and the highest HDL in their plasma. And what we can see here is that they also have their microbiome also contain a higher abundance for the potential biosynthesis of the vitamin B complex. And what we can see in the literature is that the higher the vitamin complex in the plasma of people, the higher HDL they will also contain in their plasma. We can see it the other way around as well when we look at the short-chain fatty acid metabolism. So what we see is that the group C2, which is the one that contain a lower proportion of a lot of enzymes like butanol dehydrogenase, for example, these are enzymes that do not favor the production of short-chain fatty acids. So what we see is an inverse correlation. So C2 cluster, which has a lower total cholesterol and also the second lowest BMI, they contain the higher production of short-chain fatty acids that we can see also in the fecal samples. And these results, there's not too much information out there in the literatures, but we can see it at least in few of those ones that our results also correspond to whatever have been seen before. Finally, we can also link these metabolism of short-chain fatty acids to and vitamin B biosynthesis to some metagenomic assembled genomes, and we can see that wherever they match in terms of all these genomes that we managed to screen, they correspond to what we're expecting in terms of abundances of short-chain fatty acids and the biosynthesis of vitamin B. And on top of that, we have a high correlation of close to 0.5 in only a few samples. That if we consider that the microbiome, 58 samples we have a correlation value of 0.5 in relation to the concentrations of HDL. So that's actually quite neat if we compare that we don't have a massive data set at the moment, at least with only the metagenome. So as a conclusion, we can say that a large superpopulation of LPDs in terms of HDL2A and 2B, for example, seems to drive current cholesterol recommendations. We confirm that variations in LPD subtraction correspond with parallel differences in gut microbiome composition. And we also provide new evidence that suggests that gut microbiota members need to biosynthesize bioactive molecules as vitamin B, for example, and short-chain fatty acids. And this may be required to modulate the lipid metabolism in our body. So just to finalize, this is the Counterstrike team in Copenhagen with Dennis Nielsen, Søren Balling, and Beksot Kakimov here in the Department of Food Science, and some other collaborators that we have at the University in Amsterdam, Broker, and also Unilever.